So we can just kind of keep on keeping on. So this is the last section in viruses, but again, I really want to, I put it in viruses for a couple of reasons. One, because I don't know where else to put it. Two, because that's where your textbook puts it. But prions are not viruses. So remember, a virus, in order to be like a full virus, it has to have nucleic acids and a protein coat. That's the minimum you have to have for a virus, right? The nucleic acid surrounded by the capsid, the protein coat. You can have extra features like an envelope, but bare minimum, you have both of those things. With a prion is an abbreviation for proteinaceous infectious particle. So it is literally a single protein that causes disease. There are no nucleic acids. It is not a cell like a bacterium. It is a single protein. Prion diseases can be sporadic, meaning you can just sometime during your lifetime be very unlucky and develop one. There are some that have a genetic predisposition, so you could have inherited the propensity for developing a prion, or they can be acquired, and that's generally through consumption of a contaminated product, but it could be blood-borne as well. There are lots of different kinds of prions. For the purposes of this class, and probably the way that you've heard of prions in the past, is as disease-causing agents that affect the central nervous system. And for the most part, every different kind of prion, right, from whatever source you got it, causes what we call spongiform encephalopathy. It causes holes to form in the brain, basically. And so what you have on this chart, this is from the older edition of the textbook, but the newer edition has the same table, just formatted slightly differently, are the comparisons between a virus and a prion. And really, kind of the only thing that they have in common, there's a couple of things they have in common. One, they're filterable. They're really small. So if you try to filter something that has a prion or you filter something that has a virus, they're so small they can usually pass through the filter. Right? Cells, like bacteria, get stuck. They have proteins, right? Viruses have a protein coat. Prions are a protein. And the only other thing that could, in theory, be common is an incubation period. Some viruses have a very long incubation period. HIV, rabies. Prions also usually have a very long incubation period. But pretty much everything else is different between a virus and a prion. And the really dangerous thing about prions, which you can kind of see here in the middle of the chart, how do you get rid of them? They're resistant to formaldehyde, they're resistant to proteases, they're resistant to heat, they're resistant to radiation. Okay. So they're very, very hard to get rid of. Now in humans, we call different prion diseases different names, even though they're all technically caused by the same protein. It's more about where did you get the infection or how did you get the infection. So Kuru was the prion disease that we saw in people in Papua New Guinea because they practiced something called funerary cannibalism. They ate their dead. It was a sign of respect. And if somebody died of a prion disease and you ate them, you were then exposed to the prion disease. And so Kuru is their name for shaking because people who were infected would start to have seizures. Creutzfeldt-Jakob is usually what we call it if you've eaten something contaminated, like an animal. Variant Creutzfeldt-Jakob is spontaneous. Fatal familial insomnia is inherited. Okay, and some of these others are sporadic. In animals, in sheep, they call it scrapie. like sheep and goats, because they will just tend to rub themselves, like on a barn wall, on a fence post. They'll just rub their skin raw. There's a form in mink. They don't get a fun name. They just get transmissible mink encephalopathy. In cows, they call it bovine spongiform encephalopathy or mad cow disease. In mule, deer, elk, they call it chronic wasting disease. So, but these are all caused by the same protein. Just how you get it, the form of it is a little bit different. So I tell you, it's all the same protein. So the protein is called PRPC. That's the normal form, prion protein cellular. So it is named after the fact that it causes disease. It is not named for its actual function in cells. And we are not 100% certain what its true function is in a cell, but we know that it is this protein that causes the issue. There are two forms. There's a cellular form, which is denoted by the superscript C. And there's the abnormal or the prion form that is denoted by the superscript SC, scrapie. It's the prion form. So the normal form just has more alpha helices, so it'd be like a little bit more of a flexible type of protein. If you picture secondary protein structure, what do alpha helices do? They can provide some flexibility. The abnormal form, the scrapie form, has more beta sheets, so the protein can pack really tightly together. And when it does that, when it packs really tightly together, it forms plaques. And those plaques in the brain are the spongiform, right, encephalopathy. Okay, so I'm telling you all of this, that it's the same protein, just folded two different ways. One fold is bad, one fold is okay. Yes?
So if someone has the PRPC, they don't get the disease?
Everybody has PRPC.
Okay.
So everybody, in theory, is at risk of the disease, because this protein is found in lots of cells, especially in our nervous system. Everybody has it.
But it's only considered a Prion disease if it's the folded.
That's
normal.
correct. So you only have the disease if you have the misfolded version, the scrapie version. So the problem is that when you have the misfolded version, the scrapie form, it can convert the normal form into the misfolded form. It basically says, hey guys, let's all be naughty together. And then they all start to misfold. And so that's why it spreads throughout the body. It can basically tell the normal form to misfold and become the scrapie or prion form. So in its normal form, the cellular form, the protein it's a monomer. It is able to be digested by protease. We don't find the normal form in the plaques in the brain, they call it the scrapie fibrils. It tends to be located only in the plasma membrane of cells, and it turns over within hours, meaning the protein gets recycled, right? We recycle our proteins all the time. Now, the scrapie form tends to form multimers, so it forms clumps and clusters. It is not digested by protease. It is found in the lesions. It can be found in the cytoplasm. It can be found extracellularly where it starts to form plaques. And it is very long lasting. It doesn't get automatically turned over in the body. And that's it. That's how you get disease. One bad form converts all of the other ones. So, yes.
Is there like an agreed upon method of which it actually does that? Does the abnormal form act as like an enzyme? Does it catalyze other things? Is it like a post-translational thing, or?
It's post-translational. It's probably acts like a chaperone, where chaperones are molecular folding proteins, so it's likely that. Yeah. So infection is generally through ingestion, breaks in the skin, or it could be direct into the brain. This is a risk, especially in surgical environments. And the reason that it's a risk is because oftentimes the neurodegeneration in people, it looks a lot like other neurodegenerative disorders, like Alzheimer's, Parkinson's, Lewy body dementia. They all look sort of similar if you're just kind of looking at the brain. Even the symptoms are very similar. And standard sterilization of surgical tools does not necessarily get rid of prions or denature the prions. What will happen is that the prions will accumulate in your lymphoid tissue that has nerves, and it will travel up the neurons eventually to the brain. Now, the challenge is that you don't know you have it. There are no symptoms until it's too late. It does not cause any CPE. It has a very long doubling time, a very long incubation period, decades. It can be decades. Eventually, when it gets into the brain, it can cause the neurons and cells of the brain to form vacuoles, and it forms these amyloid-like plaques. So I know we don't have a neurology course here, but maybe in some of your courses you've talked about some of these neurodegenerative disorders like Alzheimer's, Parkinson's. They're often characterized by plaques in the brain, and prion diseases do the same thing. Eventually, you'll have a patient that loses muscle control. They may have like uncontrollable shivering and shaking, dementia. These are host proteins. They are your own proteins, so they are not antigenic. You can't do a Western blot and look for, the prion form unless you do a modification of a Western blot. I'll tell you in just a minute the new diagnostics for it. They don't cause inflammation. So there's really no immune response. There's no interferon production. It's very insidious. So this is Creutzfeldt-Jakob transmission through transfusion. A 30-year-old man came to his doctor because of fatigue and inability to concentrate. They thought he had a respiratory tract infection. Neurological exams at this time were normal. History was significant for the fact that during a surgery 7 years ago, he had received packed red cells, including blood from a donor who died a year later with Creutzfeldt-Jakob disease. Within 6 months of his initial presentation, he had difficulty maintaining balance, a tendency to stagger, memory problems, a tremor in his hand, and pain in his legs. At this time, there was no evidence of changes in vision or mental status. After another 6 weeks, his mental status and memory decreased, balance and walking became difficult and painful. Imaging and EEG indicated changes in the brain. New blood tests showed the presence of the variant protein. The mental status and physical ability continued to decline. He became mute, bedridden, non-responsive, and he died 8 years and 8 months after the transfusion. Western blot of autopsy samples from the brain and tonsils contained the prion form of the protein, and I'll tell you how you have to modify the Western blot to do that. Plaques and spongiform encephalopathy were noted in the brain. Because of the long incubation period for prion diseases, prevention of transfusion transmission of Creutzfeldt-Jakob is difficult. Variant Creutzfeldt-Jakob has a more rapid onset of disease. In this case, shows the classic presentation through the 5 stages. Incubation, 6 years. Fatigue and difficulty concentrating, 18 months. Progressive neurological decline, 9 months. Late neurologic phase, 4 months. Terminal phase. Immunoblot analysis of treated prion protein can now distinguish the altered form from the normal form in samples that can be taken from the tonsils or the brain. So the way we modify that, you have to modify the Western blot. So you have, I can't draw, but whatever. So let's say that's the prion form, or the normal form, right? And here's the prion form. And I don't know off the top of my head the exact size, but I'm just going to say, let's say the proteins are each 50 kilodaltons, right? They're the same size. They're the same protein. They're just folded a little bit differently. So if you were to do a Western blot, right, here's your ladder. This one is 50 kilodaltons. Here's the C form, the SC form. They look the same. They're the same size. They're going to run out the same size on the gel. But what you can do, digest with protease. This one will not digest, right? It's protease resistant. But this one will. So now when you run the cellular form, you have all these little small fragments, but the scrapey form is the same size because it's protease resistant. So when you do the Western blot, you have to have a control of what the normal protein would look like if it were digested. And then you could have your patient sample. And if you still have sample up here at the size of the protein, you know that that patient has the disease. So you have to modify the Western blot. You can't just look for the protein. You have to do a protease digestion and see if you get a lack of breakdown. So that's how you can diagnose it. And that's a relatively new diagnostic tool. Prior to that, it was, we think you probably have a prion disease, and you're probably going to die soon, and we'll know for sure that you had a prion disease after you died. Yes?
So you can do this Western blot, like, I don't know, how early on after exposure will you get these results?
Well, you wouldn't be able to even think about doing it probably until you started to have some neurological symptoms. Mm-hmm. So yeah, it's going to be probably 12 to 18 months before death that you would be able to do that. And the problem is there is no treatment, there is no cure. So it is 100% a death sentence. And again, it mimics a lot of the things that we see with Alzheimer's, Parkinson's, other neurodegenerative conditions. But, you know, we talk about patients with Alzheimer's and Parkinson's that live for years or decades after their diagnosis, right? Often it's a very slow decline. This is very fast. 1 to 2 years after symptom onset, the patient will be dead. Okay. So I don't know. It's like do you want to know? I mean, none of the other neurodegenerative conditions like Alzheimer's, right? Parkinson's, there's really no cure. But incubation period of up to 30 years. For most patients, death is within a year once symptom onset starts. So again, they're a little bit scary. So there are some novel diagnostics that have been developed, modifications to MRI, looking for elevated levels of the PRP protein, but this is the best one, doing the Western blot. It's one of the easiest. You can also do in vitro aggregation assays. Can you get the protein to cause aggregation? Because if it aggregates like that, that means you have the bad form in there. So there are some options for diagnostics. But again, there are no treatments. It is a death sentence. So how do we prevent prion diseases? Well, if they're spontaneous or they're inherited, there's really not, you don't have much of a chance, right? But some methods that have helped, don't eat dead people, right? End cannibalism. Don't eat live people either, just don't eat people. It helps to not eat people. Remove animal products from livestock feed. If you've ever donated blood, it's been a while since I've donated blood, but the last time I did, there's a question on there. Did you live in the UK in the 1980s? Because there was a huge outbreak of mad cow disease. And so these people, theoretically, could still be incubating prions all these years later. But what they were doing was when cattle would die, they would grind them up and feed them back to the cows to give them extra protein. Yeah. So don't make cows eat cows. Or any other, like, cows aren't carnivores. They're fine. And sterilized surgical tools. So autoclaving for at least 1 hour. Bleach, sodium hydroxide, like higher concentrations than you would normally use. Honestly, I would throw out my tools if I thought, oh, this person has a prion disease. I would throw them out. But yes.
Do they like test when you do like blood and like organ donations if the patient has prions?
I don't think it's a common thing that they screen for, no. It's very, yes, very rare.
Is there anything linked to the sporadic onset of it? Or is it just one day?
Just bad luck. Mm-hmm.
I have one day.
I hope not. But in theory. Any other questions about prions? So again, they're not viruses because they don't have any genetic material. But a few years ago, there was a pretty decent outbreak of chronic wasting disease, which is the one that in mule, elk, and deer in Wisconsin. And so people that were hunting had to get the animal screened before they could take it with them. Because if they took it home and tried to eat it without getting it screened, they could develop prion diseases. So there was a big deal in Wisconsin about that. Okay, so let's move into the fungi, fungal biology. The study of fungi is called mycology. I actually know a mycologist. He's a fungi. He's kind of weird actually, but I like him. He's a cool guy. We don't have one here, unfortunately, but it's a really interesting field. You could do, entire courses on mycology. You could do a whole course on medical mycology. You could do them on environmental, ecological. Fungi play a hugely important role in the ecosystem. But of course, we are more interested in how they cause disease. We are taking medical microbiology. Fungi are our first eukaryotes that we're talking about. They're heterotrophic, right? They don't make their own food, they consume food. Some common fungal infections are listed here. We're not going to talk about all of them. We will talk about some, the ones that are more likely to be seen, especially in the US. So in the environment, they can be saprobes. They eat decaying material. We can find them as symbionts. Something like lichen, for example, is an association between fungi and often photosynthetic bacteria. So they live in a symbiotic relationship. They can be commensal, and of course they can be parasitic. They can cause disease. In humans, fungal infections are not terribly common. And most of the time, we see fungal infections as opportunistic infections. It is relatively uncommon that a fungal infection in an otherwise healthy individual is going to cause disease. Of course, we have one of the most common fungi that does that as a resident of the Central Valley, but it's not as common as opportunistic infections. Now, depending on the morphology and this is, in humans, and in the laboratory setting, you can have them be a yeast or a mold. And so a yeast is going to be a unicellular, and a mold would be like a multicellular form. Some fungi that are found as yeast will be found as yeast at different temperatures. They generally will replicate by budding or binary fission. So budding is kind of cool. Like binary fission is a lot like what bacteria do, right? They double everything and then split. Budding is kind of cool. It's like the new the new cell kind of like balloons off the old cell. And fungi have a cell wall, which is illustrated here, and you can see the scarring left by the cell like coming off under electron microscopy. So they do have a cell wall, right? They are eukaryotes, so they have a cell membrane, mitochondria, Golgi, ER, nucleus. They look a lot like a standard eukaryotic cell. Molds are usually multicellular. They form what are called hyphae. They're like projections, and they form a mat called a mycelium. So sometimes what you might see in a clinical setting is there are some bacteria, especially things like actinomyces that cause like oral infections. It's got that myces in the name because people thought it was a fungus for a while because of the way it grows. It grows a lot like a fungus in a mat. It's kind of fuzzy and fluffy. So that's what you're looking at. And they can reproduce by spores, and they can reproduce sexually or asexually. They do what they want. Some fungi can be dimorphic, so they'll go back and forth between different like yeasts, elongated forms. They do what they want. So here is some of the structures that you can see. You have binary fission, budding, these long structures. They can have, their hyphae can be septate, meaning it's like individual cells, or it can be without the individual cells. So they do a lot of different things. Fungi just can look different depending on where they are and what their life stage is. When we talk about fungal infections, they're called mycoses. So fungal infection is a mycosis. They're really commonly are going to be superficial. They are superficial of the skin and hair. They are not destructive. They don't destroy tissue. And they're generally only of cosmetic concern. Often they cause pigmentation changes. Doesn't hurt, not gonna kill you. It's purely cosmetic. Probably more commonly, the superficial are often seen more frequently in like tropical areas, tropical environments. More commonly in the US, for those of you who might be practicing medicine, you might see cutaneous infections. They affect the keratinized layer of skin. They can infect hair and nails. Really, the major host complaint would probably be itching and the skin peeling. They may have broken hair or discolored nails. This is going to be like athlete's foot, like jock itch, the ringworm, toe fungus. Yep. Yeah, nail fungus. So a lot of times they're actually treatable over the counter, except for nail fungus. Sometimes the only way, because that's when like the nail is growing, the fungus is growing between the nail and the bed of the nail. So sometimes the only way to get rid of that is to actually remove the nail bed, or the nail surgically to treat the nail bed.
I thought there was medication that you take over the counter.
Sometimes it
not
works.
Not over the counter, but you get it prescribed.
It can work sometimes, and sometimes they'll have like a lacquer that you can use to paint, like a nail polish, where it kind of soaks in. And sometimes you can take an oral to hopefully get up in there, but sometimes that still doesn't work, and they'll have to surgically remove the nail to get the infection out. I'd rather have athlete's foot any day of the week. It's the same family of fungi. There's like 3 genera of fungi that are really common that cause cutaneous infections, and they can get kind of in any of those places. Much rather have athlete's foot than to have it on my nails. No thanks. There are some subcutaneous infections. These tend to remain localized. They are more severe than a cutaneous infection. So you may have some abscesses, ulcers, but they generally don't go systemic. Then, of course, we have the endemic or systemic fungal infections. These are true pathogens that can make otherwise healthy people sick. Most often, these endemic or systemic, either term can be used, originate as an inhaled infection. They get into the lungs and then they disseminate throughout the rest of the body. And aside from your cutaneous infections like athlete's foot, the other really common type of fungal infection that we'll see are the opportunistic fungal infections. These are when often it's a fungus that's normally part of our flora. I guess you could call it the mycobiome instead of microbiome. Get to overgrow. Really commonly, like yeast infections, thrush. Okay. In people who are immune compromised, especially if they have issues with the lungs, you can get aspergillus in the lungs and it can form little fungal balls in the lungs. So those are the main categories of fungal infection. So there are very few species that are able to be what we call primary pathogens. They can infect a healthy host and make them very sick. Generally, they will possess specific virulence factors that allow them to do this. When we get to the systemic fungi, we'll talk about them and how they do that. The one we have in the valley causes San Joaquin Valley fever. It's Coccidioides is the genus. It is the only one we have on the West Coast that does this. There are 2 others that are found more eastern, but Coccidioides, valley fever, relatively common, very dangerous in this area. The primary pathogens will have 2 phases of growth. They'll have a saprobic, multicellular kind of stage in the soil, and they'll have like a unicellular parasitic phase once they get inhaled. So you kind of inhale the spores. And so what happens when I teach a class like this, I know a lot of the students have taken classes like ecology and they're like, oh, who cares about ecology? Because I want to be a doctor and I don't care about ecology. But if you follow ecology and you know what's going on in the environment, it can be very helpful because you can predict when these things are going to pop up. When you have had a very, very rainy winter and the soil is damp or moist for a long time, these fungi grow really well in the soil. And then what do we often have here after a rainy, rainy winter? A hot, dry summer where the spores get aerosolized, picked up, and people get valley fever. And you can predict it. You can see the cycle. Really rainy winter, hot, dry summer, here's everybody with valley fever. So just watch, they're predicting an El Nino this year. El Nino, hot, dry summer, fall of 24, valley fever. Call it like I see it. So ecology can be really important sometimes. And with climate change, when we have more extremes, right? Really, really rainy winters, really, really hot, dry summers, you're probably going to see this even more. So some fungal virulence factors are listed here. Of course, when we talk about the various fungi, we'll talk more about their specific virulence factors. But like everything, every other infection we've talked about, their ability to evade the immune system is really helpful, stealing host nutrients, enzymes, or proteases that can destroy host tissue, right? All really common motifs that we see with fungi. And again, as we get into the very specific different types of fungi, we'll talk more about how those cause disease.
Are fungal ear infections from swimmer's ear
Mm-hmm.
cutaneous or?
Generally, yeah, that would be like a cutaneous. Yeah. Because you're having the irritation, you're having the swelling, and I should have included it. Maybe I'll try to remember to include it later. But I have a picture of somebody that has swimmer's ear caused by a fungus, and you can actually see like the fungal structure like growing.
I think I actually have a picture of it.
Yeah, gross. Thank you. We'll look at it later.
Because I had a fungal ear infection.
I was like, why would you take a picture of that?
Yeah, you can see like, it's probably aspergillus because you can see the stalk with like the little black knot on the top, which is aspergillus, but you can see that growing in someone's ear canal. When they get it in the lungs, you see the same thing in the lungs. It forms a aspergilloma. It was your own ear infection?
No.
Oh, okay.
Please don't tell.
I was like, yeah, I don't know if I want to see your own infection, but ear, I guess, is not as bad as, somebody's like nasty wound or something. So, when we talk about diagnosis and treatment, a lot of the ways that we diagnose fungal infections are going to be similar to the ways that we diagnose other infections. They're organisms, right? We can culture a lot of them in the lab. You can do microbiology to diagnose a fungal infection. You can do microscopy. They're bigger than bacteria, so they're easier to see. And sometimes the way that they grow inside the body is unique enough to identify them via microscopy. You can do staining. You can culture a lot of them. You might have to alter your growth media. Fungi do a little bit better with a more acidic, higher sugar content. And you usually would culture them at 30. Even though the body is 37, they grow better in the lab at about 30. You can do nucleic acid testing, right? They have DNA. And you can look for their cell wall components. They have a cell wall made of chitin. We don't have chitin. They don't have peptidoglycan either. It's chitin. There are some immunological assays. So you can look for antibody production against the fungus, or you can look for fungal antigens present in the host. And you can do histopathology, different types of staining, immunofluorescence, etc. So just like we do with a lot of other infections, that's how you can diagnose them. So for any fungal infection, you have to collect your samples aseptically, transport in a sterile, leak-proof container. A lot of times, for whatever reason, with fungal infections, my watch is telling me we have lab in 20 minutes. I'm like, thank you, I appreciate that. It says you should leave now. A lot of times the samples sent to the lab are not of great quality. So you want to make sure that you transport them well. If a patient has a skin lesion or a suspected GI infection, you would probably do a biopsy. Oral and vaginal infections can be best diagnosed by clinical presentation or direct microscopic examination. When we talk about opportunistic fungi, that one will probably be Monday when we come back from Thanksgiving. So you won't have to have the mental pictures of it while you're at Thanksgiving. With the oral infection, thrush, you're literally looking at a white overgrowth in the mouth. It's very, very noticeable. And you would have, with the vaginal infection, kind of the same thing, a white overgrowth, discharge. It's pretty obvious what it is. Yes.
Is it the same type of yeast?
Yes, it's a Candida albicans usually is the one that causes both thrush and like vaginal yeast infections. It's just and the reason is because it's a common resident of the microbiome. It just lives normally with us and is not that big of a deal. Thrush is really common in infants because they don't have a very good immune system. Yeast infections, like the vaginal yeast infection, is relatively common after antibiotic therapy because antibiotics can disrupt the normal vaginal microbiome. In an adult, to get thrush, like in the mouth, is often a sign that there's something wrong with the immune system. So it's not good for the patient, an adult patient, to have thrush. You can do direct microscopic examination of clinical samples. You can see yeast, you can see hyphae, and if you see them where they're not supposed to be, or you see them in very high numbers, you can be pretty sure that it's a fungal infection. And again, some of them are so distinctive, and those of you who become clinical laboratory scientists, you will get training in recognizing these, that you will be able to look at the scope and say, oh, it's this. Right, which is more challenging with bacteria because you look at a bacteria, you're like, it's a gram-negative rod. Right, that's okay. Is it E. coli? Is it Pseudomonas? Is it Salmonella? It's a gram-positive coccus. Well, staph, strep, right? It can be more challenging with bacteria. But a lot of the yeast and hyphal structures can be unique to species. Culture takes a little longer than like some classic bacteria, but mostly we're talking about days. Not many fungi are going to be up to weeks. So again, you can see the different structures. You've got yeast and hyphal forms. These are mostly like yeast forms. Here's some hyphal forms. So different types of staining. There's like immunofluorescence, silver staining, right? Whatever process will depend on what type of sample you have. And again, that will come with experience in the lab. You'll know, oh, we suspect a fungal infection in this type of tissue. We'll do this type of staining for it. Culture is a good way to ID fungal infections. You can use a selective and a non-selective media. Sometimes they require blood for the extra nutrients to grow. If you are culturing someone's blood, there should not be fungi in it. If there's fungal infection in the blood, that's a problem. But bloodstream infections caused by Candida albicans are like one of the top 5 like bloodstream infections that hospitalized patients get, because it's their normal flora and it can get into the bloodstream when patients have like PICC lines or anything like that. So most fungi grow best at a little bit lower than body temperature, which I know seems counterintuitive. They're causing a body infection. Like, why are we culturing them at lower temperature? They just grow faster at the lower temperature. They can still grow at body temperature, but they're going to grow best room temperature. 5 days to 2 weeks is pretty common. Okay. So again, you can do antibody or antigen detection. A lot of the antibody tests we have are not necessarily sensitive for most fungal infections, and here's why. 1, some of the most common fungal infections you're going to see are things like athlete's foot and jock itch. They don't get into the bloodstream. You're not going to mount an immune response against them, right? If you have athlete's foot, at the worst, it might be kind of itchy and maybe a little bit red, but you don't get a fever. It's not swollen. There's not pus. There's nothing that indicates it's a really active infection except for, oh, well, my toes are peeling and a little bit itchy, right? So you're not going to get an antibody response against that. Some of the other most common fungal infections are members of your normal microbiome, like Candida albicans. So having an antibody against Candida albicans is not terribly informative. Or something like aspergillus, like aspergillosis. It's estimated that we inhale at least a couple hundred spores of aspergillus a day. So the fact that somebody might have antibodies against something they've been inhaling hundreds of every day for every, however many days they've been alive, not terribly informative. So it may not be informative, may not be sensitive. You can look for particular cell wall antigens or metabolites. But of course, you can do PCR to detect fungal DNA. The technology shows promise. It hasn't been fully developed, again, because fungal infections are just not as common as bacterial and viral infections. So the diagnostic tools for some of the more common infections are just more well developed. But if you find fungi in places they're not supposed to be, or you detect fungal DNA in places you're not supposed to, or you test for something like coccidioides, which people shouldn't have, right? That can be more informative. When we talk about treatment, maybe you've been thinking about this idea that we are now trying to fight a eukaryotic organism, right? Bacterial treatment is easy-ish. Antibiotics. In theory, treating a bacterial infection is easy because bacteria are so different from people. Treating viral infections is hard because in order to stop a virus, you're often stopping a host cell from replicating. So you have to identify specific viral proteins or mechanisms to try to inhibit. Fungi are also tricky because fungi are very similar to our cells. You may not think that a fungus is, similar to you, but it is. So these are just terms that, You might expect antifungal spectrum, fungistatic, fungicidal, right? We just replace bacteria with fungi, and those are the terms. But identifying the targets is trickier. One of the best targets is going to be the one feature of a fungal cell that is so different from ours, and that's the cell wall. Right? We don't have a cell wall. So if we can inhibit the fungal cell wall, that's good. So fungal cell wall inhibitors. The other really common type of antifungal is a class of drugs, or two classes of drugs, that inhibit synthesis of the cell membrane. Or at least part of the cell membrane. So in our cell membranes, in ours, we have cholesterol. So our cell membranes have cholesterol embedded in them. Fungi have a different sterol. It's called ergosterol. And the biosynthesis of ergosterol is a little bit different than the biosynthesis of cholesterol. So two classes of drugs, the azoles and the allylamines, inhibit different steps of the ergosterol biosynthesis pathway. We don't make ergosterol, so that doesn't really affect us. So those are the two best targets. We also have polyenes that cause direct cell membrane damage. The challenge here, these polyenes typically interact with ergosterol, and that's great. We don't have ergosterol. The problem is that ergosterol and cholesterol look pretty similar. So while polyenes have a much higher affinity for ergosterol, they will sometimes bind cholesterol as well. So they can cause direct damage to our cells. You get some cytotoxicity. They do mitosis a slightly different way than we do, or they use slightly different protein components so we can inhibit mitosis. Their nucleic acid synthesis is just a little bit different, so we have drugs that can inhibit that, and their protein synthesis is just a little bit different. But really commonly, most antifungals, fluconazole, right, things like that, azoles, really, really common. So Amphotericin B is another pretty common fungal treatment. It's part of this polyenes group. It binds to the ergosterol and causes a pore to form. And when that pore forms, you get the leakage and you get direct damage to the cell will die, and it's fungicidal against most fungi. So we'll stop there. What I'll post are some videos, the end of this section, and probably just a couple of the other little sections